Hello, and welcome to Giants of Gene Therapy. I'm Hans-Peter Kiem, President of the American Society of Gene and Cell Therapy. My guest today is Dr. Luigi Naldini, another true giant in our field. He has achieved worldwide renown as the pioneer of lentiviral vector-based gene therapy. Dr. Naldini is a professor of cell and tissue biology and of gene and cell therapy at the San Raffaele University School of Medicine, and he's also the scientific director of the San Raffaele Telethon Institute for Gene Therapy in Milan, Italy. For the past 25 years, Dr. Naldini has pioneered the development and the applications of lentiviral vectors for gene therapy. This has led to a number of important clinical trials for patients with genetic diseases and also cancer. Today, Dr. Naldini continues to investigate new and next-generation strategies to improve gene therapy and gene editing approaches. Dr. Naldini has been a mentor to many young scientists, and he's also a prolific researcher and has received numerous global awards, including the Grand Officer of the Order for Merit of the Italian Republic, one of the highest ranking honors in Italy, the Louis-Jeantet Prize for Medicine in Switzerland, and outstanding achievement awards from both the American Society of Gene and Cell Therapy and the European Society of Gene and Cell Therapy. Dr. Naldini has been an ASGCT member since 2000 and served on the Society's board and various committees. Welcome, Dr. Naldini.
Thank you.
Let's start from the beginning. Tell us a little bit where you grew up and about your family.
Yeah, I grew up in Torino, Italy, which is a northern industrial city. I was the first of five brothers, and my family was, of course, in good shape. I was the only one to really develop interest in biology and medicine. My father was an engineer. Most of my brothers went on to that career. And, I was fond of nature and animals, and that's when I started to really grow an interest in biology, when I started reading about molecular biology. In my high school, readings like, Jacob Monod books on DNA and RNA. That was a time when there was a discovery of biology becoming a true molecular science. That was really fascinating me, attracted to what was my then career.
So what were the next steps then, after high school? When was it clear to you that you wanted to go to medical school?
Well, I had decided, in fact, to pursue more a scientific career. I was doubting between still physics versus, biology, if you want. I was strongly advised to take medicine as the key, topic for my university studies. At that time, it was probably more common than today to take that route, even if you are primarily interested, in the science, in becoming a scientist. I'm glad I did that, but then so that was actually my next step, medical school in Torino.
And then you also did a PhD, and this was also, where did you do your PhD then?
I was joined between Torino University and Rome. And so exactly, that was when I really started to do most of my time research in the laboratory. And then I took from there, postdoctoral stay in the US. I worked with Yossi Schlesinger on signal transduction at that time. That was when we were very excited about, the growth factor receptor being tyrosine kinase and signaling, let's say transduction signaling. That was before, I became, of course, fond of gene therapy.
Right. So back in Italy still, did you ever consider or did you ever practice medicine, clinical medicine? Was that ever a consideration for you back then?
Well, of course, I did some, was part of my training, of course. I did some early work as a general practitioner, mostly I would say to, for a living. I know the fellowship early career in science were not that robust. So, but then, of course, when I moved to the US, I quit essentially any medical practice, direct practice since then.
So yeah, you initially went to Maryland, correct, as a postdoc, yes. and that was your initial work. And tell us a little bit then what you did there, and then of course the next steps when you got into vectorology.
Yeah, the early work was really in signal transduction. I had developed some, the first antibody to phosphotyrosine. So we were doing Western blot to look for receptor activity. That's what ended up really to work with Yossi Schlesinger he had moved from Israel, from the Weizmann, actually initially to a biotech entity in Maryland, and then of course to NYU, and that's where I pursued my, say, final PhD and early postdoc work. I came back to Italy as an assistant professor, setting up my lab and with the idea to continue working on that. But then I faced the challenge to really develop a research of my own as compared to what was the common research in most of the institutions over there. And reading around different emerging science, I got fascinated by gene therapy with the idea, of course, of not transferring genes. And that was really the next big choice in my career.
So yeah, tell us about that. You went to the Salk Institute then, correct?
Yes, I browsed through what exists in different institutions, and that was one, of course, of the very relevant one with early, of course, steps in gene therapy, mostly vector development. And in the Verma lab had been made. It attracted to me. Originally, my idea was to build vector which would be targeted to those receptors I had studied on. So there was a project I had proposed to Inder when I finally got to his lab. I had to queue a little bit. It was always too busy lab. And my first bench and desk was the chemical hood in Inder's lab because it was crowded, but I took that anyway. And then I found that the project I was proposing was already taken by another postdoc. And so that's when we started to discuss what to do. And then there was this idea, of course, at this point it was clearly that those gamma retroviral vectors were very relevant as tools, but also very inefficient in most of the cell type because of the requirement for cell division. And there was upstairs work from Didier Tronon's lab on HIV, which was discovering nuclear import of HIV proviruses. And so that was the idea, say, what if we could, to a certain extent, of course, ride this deadly virus, very fearful, of course. But on the other hand, could we derive an integrating vector of much more efficient capacity? So that was what really set me on the track towards lentiviral vector development.
This was also, of course, great. I mean, initially, I think you were, as you said, hoping to get better transduction modification of non-dividing cells, which was, of course, great, with the lentiviral vector. But then really you kind of almost were able to foresee what was coming. You then also worked on the improved safety features. And maybe, can you tell us a little bit about how that came about with the self-inactivating construct?
It's sort of odd. We, of course, when I was joining the field and working on lenti, as you said, the key driver of that work was making efficient vector. The concern about genotoxicity was less clear. There was, of course, knowledge of the potential risk, but there was also widespread assumption that a single hit of a vector would not be enough to realize that risk. Unfortunately, it was not true, as we all know. And so after a while, when we were already ahead in vector development, the news from the first leukemia originated from previously treated gamma retroviral gene therapy patient came out, and there was a big scare about the genotoxic risk. I think we were, to a certain extent, lucky, if you want, because out of the concern of working with HIV, which was a pathogenic virus, we had done as much as possible to dismantle the viral genome, to inactivate it, take away as much as possible from what was not needed, including the self-inactivating design, taking away those enhanced and promoted region in the LTR, which eventually were identified as key driver of genotoxicity. So I think it was a sort of, if you want, a byproduct of that effort to make a safer vector from the standpoint of replication competence, which end up also to impact on the genotoxic risk, which we prove, of course, after a couple of more years of work. Also, when I was back in Italy, to the work of Eugenio Montini in the lab, we really show it was, in fact, a safer vector, paradoxically, if you want, a safer vector.
Yeah, that was such a critical, development and advance at that time that you were able to do that. I mean, that pretty much saved the field, at least at that time.
We had a lot of time available because, as you said, the whole field was sort of on hold. And I think one of the key, idea there was that, because there was been a sort of, if you want, a failure to recognize the genotoxic risk in earlier studies, it's difficult, of course, to see leukemia in mice by, the low number of cells, the limited time of observation. So with Eugenio, we reasoned that we would use instead the tumor-prone mice, essentially to increase the sensitivity of the assay. And then we proved that there was this higher incidence, faster incidence of tumor when there was the gamma rate of transduction versus lenti. Was not initially received so well because people were telling us, well, all your mice get tumor anyway, so how can you say that your vector is safer? And then that argument was, of course, well, we knew the system is going to give tumors, but what we are looking at is the difference in timing. So right. I think that observation eventually held true in the clinic, luckily. So
yeah. And when did you decide then to return to Italy? What was that process for you?
So that was, of course, a few years after the Salk. Actually, I was originally planning to go back to Italy. I was on leave when I was on the Salk. But then, the technology we had developed at the earlier stage was licensed by a biotech in the Bay Area with the idea to further develop that. And they were, of course, also looking to scientists to take that project on. And I thought at that time that should be me. I mean, I say, why should I go back to Italy unless somebody else working with that? So I remember I called my wife. She was back in Italy already with my kids at that point. They said, well, I cannot come back. I have to go to San Francisco on the other end. And that was another couple of years in the biotech when we did develop the technology into what is the current third generation. We did all the assay and so. And then actually, at that point, the scare about the genotoxicity issue came out. Mm-hmm. So in the company, not as usual happens, from overnight, there was essentially the idea of closing those projects. The CEO of the company said, this is too scary for us to go on. At the same time, I was already ready to go back to academia. I thought my main role in the company was almost done. And so I did take, there was in the university, I had, of course, I was not anymore on the hired by university, but at that time there was a new opening for a professorship, so I took that and went back to Italy.
And that was then what year did you go back?
This was after 5 years, and it was
I think
25 years ago.
Right? Yeah. Mm-hmm. And then you went back to Milan, and
I initially went back to Torino as associate professor, where I set up my lab working on gene therapy. We started to move on. I remained linked to the company for some time because I really wanted to maintain the possibility to bring back to Europe also the technology, which we did also together, with Patrick Aubourg in France. So we did somehow manage to move all what has been done over there. And then I started to, however, realize that it was been a challenge for me in Torino to set up the clinical translation capacity, which instead was available in Milan, the San Raffaele Institute, where they had been already working on the ADA kit, using the gamma retro. And so I was actually started to talk to them. I became, initially consultant, and then I started to move there. And then I eventually moved to Milan after 3 years in Torino, again with my family. At that time, and then since then I have been working there, where of course I found the best environment for really moving the technology into robust clinical testing, and then all the work we have been doing since then.
Yeah, you've obviously set up an incredibly successful team now. Tell us a little bit about the San Raffaele Telethon Institute. How did you envision this early on to expand it? And was it more difficult in Europe or Italy than maybe in the United States at that time?
I think in that time, the San Raffaele Telethon was quite in a unique position. I would say mostly thanks to the vision of Claudio Bordignon and also Maria Grazia Roncarolo, who there initially had set up this capacity for translational development. They really had been keen on having a strong clinical unit focusing on early transplantation and early trials, together with some also capacity for manufacturing through a spin-off at that time, which was Molmed. So there was this uniquely available expertise really closer to the laboratory space. Of course, I brought all the science and technology I've been developing. It was really a match to the existing expertise. And I think this still remains a very leading component of our institute, really putting together clinician and scientist from the very beginning of a research project, sitting at the table and having a clinician giving you the time, having to discuss early on when you have some ideas whether this would ever be applicable or not, whether this would really meet or not the need of the patient in that specific scenario. I think this is to a certain extent has been, it was clearly uniquely available at that time. And today, of course, this is what many other centers have developed as well. And I think it's a key for the success of our science, right? To have that capacity. I mean, as you also know very well.
No, absolutely. You're so right there. So you've grown to, how many people do you have now in your institute?
So the San Raffaele Telethon Institute is about 250 people. There are 15 PI with their own research units, 2 clinical research units head by Alessandro for the pediatric unit and by Fabio Ciceri for the adult, and a number of core facilities which support the translational work, really like a GLP capacity for biodistribution studies, vector integration unit for monitoring insertion, immunological core, pathology, those type of important support for enabling really the bench to bedside move of the research projects.
What do you think were the biggest successes for with lentiviral vectors, and where do you see the limitations before we go into sort of the next generation approaches?
Well, I think now we can say that they really made a hit in the clinic, mostly, of course, through hematopoietic stem cell engineering. I think the combination of more efficient and safer from the standpoint of genotoxicity gene transfer is providing substantial benefit in a growing number of genetic diseases, whether immunodeficiency, metabolic disease, hemoglobinopathies. I think that there is an established success there by long-term follow-up, more than 10 years, hundreds of patients. So this is extremely, I think, rewarding for the technology. Similarly, they have been very efficient in T-cell. Of course, in T-cell, there seems to be less of a concern for the genotoxicity side. Gamma retro are also being used, but the lenti are also providing, I think, more versatility. So that's the key places today. I think there's a space for applying lenti as well in vivo, like we originally did, by the way, looking in the brain. I think liver, muscle, and brain are there to exploit the lenti as well. Of course, we know AAV is there, but there's a lot of unmet need as well. So that's something we are looking forward. On the other end, of course, lenti integrates semi-randomly in the genome. So there's always an inherent risk of genotoxicity, despite all, I think, the design features that we have discussed. Ideally, if we could target insertion to a specific site, of course, we would have a much precise output and also potentially safer. We cannot do that with lenti. Unfortunately, we can do that with genome editing, where most of the people are doing this, of course, including us now, together with new challenges.
So do you think, will gene editing and these next generation gene editing technologies replace the use of lentiviral vectors, do you think?
Well, it may eventually. I think not now. As I said, I think we should leverage on the long clinical testing of lentivirus. So we still have to check gene editing. I mean, gene editing seems to be very promising at early results. We have to look more in the long-term follow-up if those edited grafts remain robustly polyclonal, for instance, as we see with the lenti. The other thing is editing works extremely well at the moment for gene disruption. For gene correction, we may consider a new approach like gene-based editing or so, but that's maybe limited to certain correction of a few bases. But long-range editing, integration of new, like cDNA, which is currently the most feasible approach to correction of most genetic diseases, which there are multiple mutations to correct. You cannot have a single therapy for any mutation. I mean, this remains a challenge for conventional gene editing in terms of efficacy and the outcome of the break. So lenti is still there, I think, as an alternative. So we have to work on that. That's what we do now in the lab. We work on gene editing. Of course, we hope to test in the clinic as well and to figure out what are the relative pros and cons.
So, of course, many of the diseases now we and everyone targets now, for example, diseases like sickle cell disease, they're much more common in low and middle income countries. So how do you envision that we're going to be able to bring this sort of promising gene therapy, gene editing to these people and these patients? There's about 4.5 million patients with sickle cell disease globally, but very likely less than 1% will have access to promising ex vivo gene therapy approaches.
Hans-Peter, that's really a huge issue. I think this is one of the things which really, on the one hand, we have this sort of a reward. Say we managed to develop a new treatment. On the other hand, say we do see that this is so difficult for patients worldwide to access them. Of course, as we all know, this treatment comes with some of the highest cost ever. And despite the fact that this being once and done, they could be ideally suited also to country when there is, of course, less developed health system, rather than the need of continuous replacement treatment of care, the possibility to provide once the treatment, maybe in a specialized center, and then the patient benefiting potentially lifelong, I think is ideally suited. So I think that's what we have to fight for. Whether this can be achieved ex vivo, as currently is done in the clinic, or maybe in the future directly in vivo, which is more challenging, but again, science is moving toward that direction. We'll have to see, but I think we have to start fighting for moving towards that. On the other hand, establishing centers that can provide this treatment to those countries as well. And once, I think this is feasible. And secondly, trying to work on price and access, together with charity countries, nations, to figure a way to allow this without jeopardizing, of course, innovation, and without, of course, still providing the reasonable reward to the company which are engaging into this development as well. So I think it's a difficult field. We have to move carefully. Cannot really claim there is a simple solution.
Yeah, no, I agree. How important is it, for these gene therapy studies now to really have industry involved from your experience?
Obviously, industries where you would mostly say pertains to the development of a new drug and also mostly making it available on the market, right? So in principle, that's where you should do. On the other hand, there may be specific sector where this may not be doable the way we have been doing until now, in particular for rare diseases, where most of gene therapy actually started from to validate, of course, itself. And unfortunately, after that validation, the commercial interest in those treatments becomes much, much less. Currently, despite the high costs, we are seeing companies, whether of mid-size or large size, to essentially let it off, or close those therapies for which there is too limited
access
in terms of patient need. I think we have to explore an alternative route there. Establishing center which can produce and administer in a non-profit manner those treatment to the patient in need, with public, of course, funding, with some sort of alleviation of the regulatory requirement, not, of course, to the detriment of safety, but sometimes to the detriment of what you would expect from a commercial product. So I think we have to. I think that's where the field is also moving, both in Europe but also in the US. Figuring out an alternative path, at least for those treatment and disease for which a commercial, standard commercial model of development doesn't apply anymore.
Yeah, I mean, in particular, what of course comes to mind is ADA SCID, where you guys have really shown just beautiful promise and success with this therapy, and of course Don Cohen's group and others, and then why do you think that this gene therapy was not successful within industry?
Well, because for that reason, I think the really limited now. I mean, there has been, as you know, this drug was approved in Europe as the first ex vivo gene therapy. This is still using the gamma reactor platform, but has been substantially safer in that disease as compared to other, for reasons which are not entirely clear still. and yet, those. So although we have been providing that commercial drug, still, the number of patients is too low to continue support, and also the cost of that, which was initially agreed, was when GSK, that was a big pharma. They were, I think, brave enough to negotiate a relatively low price as compared to the current standard, maybe with a view toward development of the platform towards other diseases. But then a change in the management, essentially, sell off this whole platform to a smaller company, which, Orchard Therapeutic, which has a different, of course, portfolio, and they have to benefit from every single treatment they are developing to the maximum extent. So they decided not to pursue further. Now, this treatment is back to the ownership of Telethon, which was, of course, sponsoring it when we developed. And Telethon has decided it's almost ready to do it, to continue to keep that on the market. Now, Telethon becoming a producer of the drug itself, of course, under this scenario of a non-profit organization making the drug available. So that's where, again, we are experimenting here, this novel track I was mentioning before.
Yeah, no, this is obviously very, very important. Yeah. So I'd like to shift gears just a little bit and also ask you, you've been involved in the international summits on human genome editing.
Yes.
Especially earlier this year, of course, again, you've participated. And I just wanted to get a little bit your thoughts on that. No, I think this year was mostly on somatic gene editing. But of course, you are familiar with several years back with Ha Jiankui's human embryo editing studies. And how has the field since then evolved or thought about this?
Well, I think, as you know, I think that there was more reason to appreciate and discuss the actual, I would say also encouraging development in the field in terms of application of gene editing to treatment of diseases, of course, in the somatic cell. I think that there are, as I said, the very promising early results in sickle cell for ex vivo treatment. There are expectations for editing T cell in different ways to make allogenic product, to improve CAR T. There was early data on liver, directed silencing. There's the new platform-based editing. So all of this remain, of course, a very good reason to share those results with a larger community, together with the ethical and societal issue of the access, the cost, so the challenge facing. So I think this has became the preeminent focus of the meeting. So where the field is going, and I think in terms of science, the biggest challenge that was put on view was delivery. I mean, as usual in the field of gene therapy, we always discuss delivery, right, from the very beginning. And again, delivering of editing machinery in vivo remains challenging. Of course, we can do it very well in animal models, but when you face clinical translation, this is not the same game. And so that's where I think a lot of effort are seen. We just mentioned before, gene editing stem cell, for instance, hematopoietic stem cell in vivo, of course, is very challenging because of the delivery, not because we don't have the tool otherwise. And so I think most of the summit went into that. In terms of the germline, I think, again, there was more a realistic discussion of what would be the actual way to edit germline or germ cell, or primordial, or depending where you would go, at which level of the germline you would intervene. I think there is progress there on the research side, but at the same time, there is a clear understanding, acknowledgement that no technology is ready for actual testing in a human embryo or germ cell at this stage, because of the still unknown consequence of an edit throughout the genome. We are realizing the risk. So I think that was the final message of the summit. Still remain too early to consider clinical testing of germline. On the other hand, good reason to continue and even more somatic testing of editing for different purposes, facing the challenges, but also realizing the successes today.
Yeah. It was nice to see, though, right? There was very good, I thought, international discussion and consensus on that topic.
Yes. I think, although one of the also a leitmotif of the summit is also to really bring different perspective, different vision from different culture. It's always an important, you always can learn from that, right. Learning from representative of not only of patients, but also minorities, different culture and approaches. And as you said, there are some common, of course, consensus on the general theme, I said, but when you go to more specific aspects, there are emerging interesting differences. For instance, poor attention when you work on, just to make an example, on genome databases. If you look at the representation of the different ethnic groups, this is very imbalanced as compared to what is found in the databases. Some of them, some ethnic population, which are the majority of the world population, are poorly represented in the databases. So when you look, for instance, for a potential off target of an editor, you are not seeing really represented the actual population as from the world side view. And again, this is something we should consider from the beginning. This is a simple example, but there's much more there will be much more to mention here.
Yeah. So what would you say? I mean, you've obviously built an enormous team and institute there. What do you do in your own lab, in your own group right now? What sort of excites you the most? Where do you want to go over the next few years?
So, I think, again, we still remain primarily concerned on exploiting stem cell hematopoietic stem cell gene therapy. We try to broaden its use also beyond, genetic diseases. I think there's a lot of potential there for making your cell, or maybe some of the progeny of those cells, smart delivery cargo for treating acquired disease like cancer. So that's one area. So expanding application beyond the original use, mostly in the cancer immunotherapy. And the other, as mentioned, is leveraging on gene editing approaches to really assess the power of this new technology in terms of precision. And third, I think what we are doing is trying to alleviate the burden of current stem cell gene therapy, in particular, the conditioning strategy that are required to make space for the engineered stem cell. And I think there's possibility there. There's been a lot of work around, including from your lab, and we are discussing on this as well, Hans-Peter. How can we really change the way we make space for those infused cells? And I think that's something we are working on. And I think in a technology we are leveraging on is RNA technology. I think RNA is going to transform the gene therapy field as well. It has done already, it's doing on a vaccine, right? But I think the transient delivery in a manner which has no genotoxic risk of a potentially very powerful function is all to be captured in a different area, including, for instance, for engrafting advantage, as we have been doing here, but also in many other areas. So that's what we are working on now.
Oh, that's wonderful. Yeah. So you've mentored many scientists and trainees. What would you advise young scientists or trainees, students today?
Well, of course, you always look for enthusiasm and commitment here. So I always think that this area of science we are working on, the translational, if you want, is extremely rewarding because you have a combination of basic science and discovery together with the possibility to really see the application and trying to balance the two. I think it's important to work on both sides as well. I mean, to maintain that balance, whether or not it's a trainee or even more if you are an independent scientist, capture, I think, innovation from technology, but also from science, no? I mean, not be blind continue to be open and interested in the broader science development, where I think the solution to your problem may come, some way inadvertently. At the same time, maintain, if you are in this field, a careful attention to the final, translation of a clinical outcome, not to take unrealistic route. So as usual, I would recommend focus on a project with this strategy of being open to, I say, scientific knowledge, innovation, but at the same time focus on your experimental strategy and keeping in mind the ultimate goal. Is it realistic? And checking this very so often, maybe not yourself, but also discussing with others. Am I doing something which makes sense? Would this be ever be able to be translated into some clinical therapeutic approach. I think to maintain that from the very beginning, this sort of a balance, science and development, translational and innovation is where we can do the best, science and career.
Great, yeah. So with your busy schedule, with your institute, your lab, you're traveling, do you have any time to do anything in your spare time?
Yeah, of course, I try to say some of that. I think it's important for your health, for your brain function as well. I always try to, and I think, I do find actually fun in a lot of other things. I like to of course, to read, to listen to music. I'm very fond of art, contemporary art. I like to go to fair. I'm also an enthusiastic collector. And art, that's where some of my earning goes sometimes, but I think it's very nice to go, but not to really see other manifestation of human creativity. Of course, and art is a primer there. Music as well. And music.
Any particular artists?
Well, I like a lot. Of course, I know most Italian contemporary. It's a European contemporary art. I'm very fond, of course, of abstract expressionism. Not easy to collect at this point. This is, of course, so you have to go into the more nuance. But I think there are paintings, are always a sort of back now in the interest. There's been a time it was like installation, big museum structure. I think now we see back interest into more the actual painting, which is more affordable to enjoy and sometimes to collect. And the art scene is very, very lively. Of course, here also from different countries and culture as well, although I am more based on European American, I would say.
Oh, wonderful, yeah. So anything else I should have asked, Luigi?
Oh, I think we discussed a lot. I look forward to the upcoming meeting, right, in Los Angeles.
Absolutely. Very much looking forward to that and seeing you there. Yeah. So thank you very much, Luigi, for taking the time today. Sure. And see you next week.
Thank you, Hans-Peter, so much. Bye-bye to you and everybody who has been listening to us.